The title of this practical is Identification of Leukocyte and Differential Leukocyte Count. First we did total leukocyte count, that was TLC, and now we are going to do differential leukocyte count. We know there are about 5 types of LBCs, that are neutrophil, eosinophil, basophil, monocyte and lymphocyte. Neutrophils are the most abundant, and these are the first cells that respond to an infection of bacteria or viruses, and they are about 70%. And eosinophil, they are about 5% of WBCs. They fight off bacteria and parasite worm, and they also bring allergy symptoms. Basophils are least, they are 1% of WBCs, and they give non-specific type of immune response to pathogens, and they are also responsible for the onset of asthma. They release histamine that causes the inflammation in the tubes of the respiratory tract, bronchioles, etc. And then there are monocytes. Monocytes are the cleaning system of the body. They are about 5 to 12% of WBCs. They migrate into tissues and clean up dead cells. Lymphocytes are T and B cells. T cells are phagocytic, and B lymphocytes, they give rise to the humoral immunity, mean they produce antibodies. These antibodies to neutralize the antigens, and these T cells are called as T because they get mature under the influence of thymus gland, and B cells, they get mature inside the bones. All these blood cells, they are originating in bone marrow, and then the T cell mature in thymus. This practical is important for the determination of type of infection, because if neutrophils are more, it means there is bacterial or viral infection. Eosinophils are more, it means there could be allergy or the parasitic worms, and if there are basophils more, it means there is some asthmatic type of symptoms. If monocytes are more, it means there are more cells that are dying, and lymphocytes mean there are some specific immune response to the antigens. For this practical, we need glass slide, staining tray, stopwatch and microscope with oil immersion objective, and then we need the chemicals, that are Leishman stain and the buffer water. Leishman stain is made up of eosin and methylene blue that is dissolved in methyl alcohol. Eosin is acidic stain and stain the cytoplasm of the cells, and methylene blue, because it is basic, it can stain the acidic part of cell, that is the nucleus. We know DNA is present in nucleus and it is acidic. So the buffer water is also required in order to provide the ionization of the dyes for staining, because these stain, when they are prepared in alcohol, they are not ionized, and when they get interact with water, they get ionized and stain the nucleus and cytoplasm. Test that is needed. The cotton swab in order to disinfect finger, and blood lancet to prick the finger, and take this blood on clean glass slides. And then we need a staining jar in order to stain the slides with Leishman stain, and is composed of eosin and methylene blue. We need dropper to apply the stain and buffer water to the slides. Then we need oil to see these slides under oil immersion objective of the microscope. We are going to take a drop of blood on the glass slide, and by using another glass slide at 45-degree angle to spread the blood drop on the slide to get a smear. So here we put this slide for drying. So we have made a few of the slides in this manner. After drying, now we are going to put these slides on staining jar in horizontal position and to apply the Leishman stain. Leishman stain is purple in color when it is non-ionized, and when we add water it becomes pinkish. We have to add 10 to 12 drops of the stain in order to cover the smear on the slides. Methyl alcohol in stain fixes blood on the glass slides and prevent washing out of the blood when we proceed through steps of washing. So we added stain and left it for 2 minutes, and then now we are going to add on each slide 2 drops of buffer water, and then we are going to blow it in order to mix it so that the stain get ionized and stain the smear. Now we put these slides in vertical manner and going to wash them with the tap water in order to remove the extra stain. Wash them thoroughly. Now we are going to wash them under tap also. After washing, we are going to keep them in a vertical manner in order to dry them. And now we can see these slides left for drying. Now we are going to see them under microscope under the oil immersion objective by using oil. The result of this practical are that we observed neutrophils that are 54%, basophils 1%, eosinophils 8%, lymphocytes 34%, and monocytes 3%. How we identified these WBCs? Monocytes and lymphocytes, they don't have any cytoplasmic granules, and the eosinophils, they have bilobed nuclei, and their nucleus appear to be in 2 parts, and they are stained with the pinkish color. The lymphocyte and monocyte, they are bluish purplish in color, and the basophils, their nucleus is stained purple and their cytoplasm is stained bluish, and they have bilobed nuclei. And the neutrophils, they have trilobed nuclei and they are bluish purplish in stain. This is all about this practical.